Helicobacter pylori infection is a significant factor in the development of stomach cancer. The Helicobacter pylori is a spiral-shaped, gram-negative bacterium that primarily resides in the stomach lining, specifically in the mucus layer that protects stomach tissue from acidic digestive juices. The unique structure of this bacterium, including its flagellum, allows it to penetrate and thrive in the acidic environment of the stomach. It is able to neutralize stomach acid by producing urease, an enzyme that breaks down urea into ammonia, thereby creating a more neutral environment around the bacteria that allows it to survive. Helicobacter pylori is a bacterium that infects the stomach and is considered one of the most common bacterial infections in the world. It is estimated that nearly half of the global population carries this bacterium, but not everyone develops symptoms or related health problems. But when Helicobacter pylori does cause problems, it can lead to a variety of gastrointestinal diseases such as peptic ulcers, chronic gastritis, and in severe cases, is a significant factor in the development of stomach cancer. The symptoms of Helicobacter pylori infection vary, and many people do not have symptoms for many years. However, when symptoms do occur, they are often related to the stomach and digestive system. One of the most common symptoms is a burning or stinging sensation in the stomach. This pain is usually worse when the stomach is empty and can last from minutes to hours. The discomfort may come and go in waves, making it difficult for some people to connect their symptoms to a serious underlying condition, such as an Helicobacter pylori infection. Bloating is a common symptom characterized by a tight or swollen feeling in the abdomen. This is because the bacteria interfere with the production and digestion of stomach acid, causing a disruption in the normal digestive process. Helicobacter pylori infection can cause excessive burping or belching because the bacteria interfere with the normal breakdown of food and release of gas in the stomach. This can cause discomfort and embarrassment and is often accompanied by bloating. Nausea is another common symptom, with many people reporting stomach discomfort that may lead to vomiting. While vomiting is less common, persistent nausea may be a sign of an Helicobacter pylori infection. Persistent stomach discomfort may also lead to a loss of appetite. Pain or bloating when eating can make food less appealing, leading to a decrease in food intake. Unexplained weight loss may occur due to decreased appetite and impaired body's ability to absorb nutrients caused by Helicobacter pylori-induced gastritis and ulcers. Heartburn or a burning sensation in the chest or throat may be the result of acid reflux. Helicobacter pylori disrupts the balance of stomach acid, causing overproduction of acid, which can back up into the esophagus and cause discomfort. In some cases, people infected with Helicobacter pylori may notice that their stools appear black or tarry. This may indicate bleeding in the stomach or intestines, often due to ulcers caused by the bacteria. In rare cases, vomiting blood or vomiting coffee ground-like material may occur, indicating upper gastrointestinal bleeding. This may be a sign of an ulcer caused by Helicobacter pylori or more serious damage to the stomach lining. How does Helicobacter pylori cause stomach problems? The pathogenicity of Helicobacter pylori is associated with several mechanisms that allow the bacteria to survive in the acidic environment of the stomach and damage the protective lining of the stomach. These mechanisms may lead to inflammation and ulcer formation, ultimately increasing the risk of stomach cancer. Helicobacter pylori has several adaptations that allow it to survive the highly acidic environment of the stomach. One of its most critical survival mechanisms is the ability to produce urease, an enzyme that breaks down urea, a substance naturally present in the stomach, into ammonia and carbon dioxide. Ammonia neutralizes stomach acid, allowing Helicobacter pylori to create a less acidic microenvironment in which it can survive. Once Helicobacter pylori colonizes the stomach, it begins to destroy the mucus layer that protects the stomach lining from digestive acids. This destruction allows stomach acid to penetrate the stomach lining, leading to irritation, inflammation, and eventually the formation of ulcers in the stomach or duodenum. Infection with Helicobacter pylori triggers an immune response in the body that leads to chronic inflammation of the stomach lining, also known as chronic gastritis. Over time, this inflammation damages the epithelial cells of the stomach and weakens the protective mucosal layer. This chronic inflammation is a key factor in the development of peptic ulcers and a precursor to more serious diseases such as stomach cancer. Studies have shown that Helicobacter pylori causes genetic mutations and epigenetic changes in the cells lining the stomach. Some strains of Helicobacter pylori produce a virulence factor called cytotoxin-associated gene A, which can disrupt normal cell signaling pathways, leading to uncontrolled cell growth and division. This process can lead to intestinal metaplasia, precancerous lesions, and ultimately stomach cancer. How to prevent Helicobacter pylori from entering the stomach, especially in areas where the bacteria is prevalent or the risk of complications is high. Helicobacter pylori is mainly transmitted through contaminated food, water, or direct contact between people. Here are some preventive measures that can significantly reduce the risk of Helicobacter pylori infection. Good hygiene habits are one of the most effective ways to prevent the spread of Helicobacter pylori. These measures include washing hands thoroughly with soap and water after going to the toilet and before eating can help prevent the spread of bacteria. Since Helicobacter pylori can be spread through the fecal-oral route, it is vital to maintain proper hand hygiene. Avoid sharing personal items such as eating utensils, glasses, or toothbrushes with others, especially if someone is known to be infected with Helicobacter pylori. Helicobacter pylori can enter the stomach through contaminated food and water, so it is important to ensure that food and water are clean and properly prepared. Cook food thoroughly. Make sure meat and seafood are thoroughly cooked to kill any potential bacteria. In areas where water quality may be poor, drink treated, bottled, or boiled water. Avoid drinking untreated natural water sources such as lakes, rivers, or wells. Raw fruits and vegetables should be washed thoroughly before eating, especially if they have been handled by others or grown in areas with questionable sanitation. This helps minimize the risk of ingesting Helicobacter pylori on food surfaces. Diagnosing Helicobacter pylori infection is essential to start treatment promptly, especially if the bacteria causes diseases such as peptic ulcers or increases the risk of stomach cancer. There are various diagnostic tests available, each with its advantages and limitations. The choice of test usually depends on the patient's symptoms, clinical history, and whether they have been treated for infection in the past. A blood test is one of the most common ways to detect Helicobacter pylori. It measures the presence of Helicobacter pylori antibodies in the blood. These antibodies are produced by the immune system in response to infection. The urea breath test is one of the most reliable and non-invasive ways to detect active Helicobacter pylori infection. It uses the bacteria's ability to produce urease, which breaks down urea into carbon dioxide and ammonia. The stool antigen test detects the presence of Helicobacter pylori antigens in a patient's stool sample. This test can be used to diagnose active infection and confirm eradication after treatment. An endoscopy involves inserting a thin, flexible tube with a camera attached through the mouth into the patient's stomach. During the test, the doctor can visually inspect the stomach lining and take tissue samples for testing. The biopsy sample is then tested using a variety of techniques, such as a rapid urease test, histology, or bacterial culture.